Okay, so let's get started. So we have started the webinar, right? People can join our journey. The webinar has started, right? Yes. Yes. Yeah, we can start. Okay, so it's really my great pleasure to welcome you to our first in-person theoretical and computational biophysics group seminar in 2 years almost. And it's really a great pleasure to have Abhi Singharoy here. Many of you know Abhi. So by way of introduction, Professor Singharoy is from the School of Molecular Sciences, Arizona State University. So he's visiting us from there. So he did his initial training in chemistry and with a minor in math and physics in India, and then joined the Indian Institute of Technology to study physical chemistry for his Master's of Science. Then Abhi moves to the United States and joins the Indiana University in Bloomington, where he did his PhD and started working actually with macromolecular systems during that period. And then I think the group was very lucky to get him as a postdoctoral associate after his PhD. So he joins Beckman Institute, the group as a postdoctoral fellow. In fact, he received actually 2 different fellowships during his time here. Beckman Fellowship that everybody knows, it's very hard to get that kind of fellowship, so it must be really good to develop those. And also a fellowship from the CPLC Center for Physics of the Living Cell, so Abhi received during his time. And then since 2017 or 18, January 2018, he joins actually the School of Molecular Sciences at the State University as an assistant professor, where he's doing extremely well, as I hear. So Abhi is extremely, I think it's an exemplary model for those of you who want to actually reach an elite state. So he's extremely active and he's everywhere, really, both on the science side and the community side, review panels, active in conferences, organizing conferences, participating in events that happen at BTS, for example, ACS, and so on and so forth. And also on the science side, he is actually very active, doing a range of projects from photosynthesis to vaccine development and also macromolecular problems that many of us will work on. He's present at supercomputing executive committees, sort of serving actually his role there and helping people to use the supercomputing the best way possible. His research is funded by multiple funding agencies already after just a few years. Actually, he has had DOE funding. He got his NSF Career Award actually just last year. Just last year it started for, which is actually a very prestigious award. NIH funding is a co-PI on a couple of NIH funds. And also private foundations, companies. So you can see, for him, I think the day is 48 hours, not 24 hours. I don't know where he finds time to do all his activities. And that has translated in a very remarkable trajectory and path that he has embarked on. So I look forward to see more success happening. So he's currently a director of the DOD Center that just started at Arizona State University. And as I mentioned, he received the NSF Career Award and also an Insight Award that you know how hard it is to get from the Department of Energy. He was also a speaker at the Future of Biophysics this year already at the Applied Physical Society meeting, which is a distinguished kind of lecture event. So he is active both on the methodology, and application side of science. That's a signature of successful people and something that you should all keep in mind, even as junior biophysicists. It's always a great idea to sort of spread yourself between these 2 aspects and combine them when possible. So we were fortunate to have him here. So I know you're very busy and have many invitations to the seminars It's very great to have you back, Abhi, here.
Thank you.
Thank you.
Thank you. I think, after introductions like this, you usually ask everyone to go home because that's the best part. But because I'm really happy to come here, also because it's home essentially, and so really wanted to share what we have been doing, and come back after a couple of years. Usually, as Imad mentioned, I'm about to go for my mid-year review in September, so it's good to kind of touch base with mentors, with colleagues, and kind of gauge what they feel about what we are doing. So thanks a lot for having me. Okay, great. So then hopefully people on Zoom can hear me. I cannot understand, of course, if you're doing it, but I'm sure that you're enjoying the talk. So with that then, let's begin. So we're going to talk about, everyone in the room can hear me, right? Right. Okay, so we're going to talk about how molecular modeling is used during the pandemic. Of course, you have had different flavors of this kind of a talk, this kind of how molecular dynamic simulations, molecular biophysics has been helping in coping with the COVID. A lot of work has, of course, happened in terms of studying what the monster is, what the demon is, like what SARS-CoV-2 is, what are the spike proteins, a little bit of work on vitamin D proteins, we are basically working on, let's say, how to come out of the pandemic. So how is the molecular dynamic simulation helping towards our rescue from the pandemic? So let's start on that. So just a little bit of an introduction to the group. As Imad had mentioned, we have been quite fortunate with the funding agencies. A number of whatever concrete major funding agencies are there in the country. They have been able to endow us with their generosity. So thank you to all of them. We have a very nice group. A number of people from the group, some of former DCBG students who are now not in DCBG, are very, very actively working with us every day, like literally as part of the group. So we are very, 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 very grateful to them. And of course, when I first started my group, we got allowed some funds, some NIH funds to be transferred. That gives you a lot of confidence in your person to start your career. Thank you, Mark, for that also. So what we have been doing is, we have a number of scientific interests. So today, because it's a science talk, I'll talk primarily about science, not too much about development. So we work a lot on molecular models. This is primarily our MSF kind of work. Structural biology, basically cryo modeling. A little bit of bioenergetic membranes, that's where the DOE comes in, NIH comes in. And vaccine design. So today, we are going to talk primarily about vaccine design. Okay. So let's move on. These are, of course, the students. We have two amongst us, Johnny and Eric. I'm sure that they might be on Zoom. And Depan is, of course, now a postdoc with Josh. And so, yeah, and then still continues to work with us. So it's fine. And Josh has no objection if you say that. So good. So then these are the 4 people who have been primarily working on this project. So, okay, let's start out. So as I said, we are primarily working on the vaccine development aspects of this. So this is a world map of the vaccine distribution. The kind of vaccines that we have gotten in this country are Pfizer and Moderna. These are 2 mRNA kind of vaccines. In fact, the vaccine that is most popularly used around the world is Oxford AstraZeneca's vaccine. It's used around 184 countries. Oxford AstraZeneca, J&J, and Sputnik. These are different kind of vaccines. This is what you call a vector-based vaccine. So an mRNA vaccine, what that does is you literally, transport the mRNA and you expect the cellular machinery to create the vaccine for you inside the cell. It's a very, very recent and of course, a very successful technology that had been, we had been working on that for 15 years. Now essentially it has come to its prime. The vector-based vaccines are literally older. They're a more traditional way of vaccine discovery. What you do is you take a virus, a harmless virus, let's say, which is not the virus that infects you. And essentially, in that virus, use that as a container, as a vehicle, to put in the genome of a particular protein that you want to, of an antigen that you want to generate. For example, in these vector with Johnson & Johnson vaccines, AstraZeneca vaccines, or Sputnik 5, we'll show you what the vaccine looks like. You basically use a particular part of the genome that encodes the spike protein of SARS-CoV. And essentially you inject that, and you expect that the outer coat or the vector itself is not going to affect you. The capsid is not going to be infectious, and the genome will create spike for you, and the spikes are going to generate immunogenicity inside the body. And there's of course a third kind of vaccine called the whole virus vaccine. This you'll find in Sinovac, Sinovac, where you actually use a deactivated virus, which of a different kind altogether, but a full virus. Let's say they use rabies virus or hepatitis virus, which is used to generate immunogenicity against SARS-CoV-2. So turns out that these are like 40 to 50% efficacy. Some of these are close to 70 to 80, and mRNA viruses are like pretty high, 90%. Good. So this was the overall world distribution, and I should say again, like 184 countries. So if you see the number of countries, purely by the number of countries, you see that the vector-based vaccines are still serving a lot of countries, including the US, because Johnson & Johnson is available. Okay. So again, as I said, like this is the form of this of an adenovirus. So what this AstraZeneca and J&J vaccines use, they use the outer capsid of what we call an adenovirus. So there are two different adenoviruses. AstraZeneca uses something called a ChAdOx adenovirus. See, and I'll show that later. It's a chimpanzee virus. That's why the CH, chimpanzee adenovirus. And J&J uses Ad26, adenovirus 26, a different variant of the adenovirus. So the primary content of an adenovirus, in the most simplistic physical way, is it has these fiber knob proteins shown in green. Again, this is a cartoon for now. Let's translate that to structure in some minutes to come. But you actually have this fiber knob proteins, which are used for the receptor. They're used as receptors. These are the ones that are used for engaging with the cell and entry. Then you have the hexon and penton proteins, which are covering the outer surface of the virus. You have a lot more of hexon proteins than the penton proteins. And if you see from the inside, there are a lot of other minor proteins that are important for the stability of this entire capsule. And then, of course, inside this is normally it's a dsDNA, it's a DNA virus. Now, this part inside is replaced by the gene that is encoding for this part. But that's a very simple-minded description of what this vector-based vaccine looks like. A very general description. Good. So now then, based on this and based on the promises of vaccines like this is not the first time that a vector-based vaccine has been used. Vector-based vaccines have been very, very popular in a number of other diseases. And therefore, when this screen sharing is not here, the number here is $1.2 billion. Yeah. So the number here is $1.2 billion. So what happened was, when the pandemic came in, US had actually invested $1.2 billion in vector-based vaccines. Now then, thank you. Yeah. So now then, what happened was, of course, what happened we all know. People started getting side effects of these vaccines. A very minor side effect, but this happened in the trials. So early on, this was found in AstraZeneca vaccines, where during the trial itself, a very rare disorder of blood clotting came out. And that right away kind of, got AstraZeneca cancelled out of this country. Despite, this big investment. Johnson & Johnson turned out lucky. So they still had the same vaccine. Now we know can actually cause similar disorders. But their patient trial group was not like, turns out that by probability, they didn't have someone who during the trial group itself had the blood clot. But they were prone to the same biophysics that would lead to blood clot. So that was their.
Same as AstraZeneca.
Yes, okay. Same as AstraZeneca. But AstraZeneca came up first because in their trial group, the probability was that someone was there, but the blood clotting happened, which in the Johnson & Johnson's trial group, it was not. So then the Johnson & Johnson got approved. But the papers have been written. Both the vaccines are implicated in virus-related side effects, vaccine-related side effects. Okay, so now I should introduce to you a key player, a key protein that is implicated in this in this entire conundrum. Right? So there's a protein called PF4 or platelet factor 4. So it's a, as you can see, it's a small dimeric protein, primarily positively charged. You can see colored mostly positive. They have negative charges inside, but the surface is highly positive. So this protein's normal function is actually as a chemoattractant. So when you have any injury, when a tissue has any injury, this protein is a part of the response machinery. It's what you call a chemokine, a cytokine, or a signaling protein. This protein actually allows for the formation of neutrophils or white blood cells. So in normal conditions, the role of this protein, PF4, is actually to build resistance against wound. Any sort of cut, any sort of wound, you want to make neutrophils, and that's what this protein does in normal conditions. However, during turns out this has been hypothesized, but actually people have found this part that during the vaccination, the same protein PF4, either by itself or after binding to some unknown factor which is not known, interacts with the antibody. What you call this is and this antibody is an autoantibody. So normally antibodies are the ones you would expect, like a virus comes in and then your body creates antibody. Sometimes the body creates antibody out of its in response to its own parts, to its own proteins. So an autoantibody, so during this virus-related clotting mechanism, this PF4 undergoes a binding with a VIP antibody. I'll tell you what VIP is. It's called vaccine-induced thrombotic thrombopenia. So it gets bound to a specific antibody, the other end of which is connected to a receptor. The name of the receptor doesn't matter at this point of time. But because of this binding process, downstream platelet activation happens. And of course, you know what is blood clotting. You have a lot of platelets coming together, unnecessarily, so to speak. So essentially, this event that is normally not supposed to happen happens when these vaccines are actually introduced in human blood. Because normally these PF4s are not supposed to be binding these antibodies. They would actually do a totally different function. They would just heal us from bleeding. But this is happening. And the question is why? Of course, this affects different places, starting from brains to basically, different organs of the body can be affected, lungs, liver, whatever. It's spread all around. And therefore, patients of BIP have reported blood clottings in not just one part of the body. They have seen all over the body. Good. So now then, of course, it's not good, but that's a mechanism to start off. So, okay, there's a risk here. How bad is the risk? So, turns out for 25 people, 25 years old around, 11 out of a million are prone to this risk. 54 years and old, it's 4 out of a million. Actually, lesser. It's not more. It's lesser. These numbers look very, in a simple-minded way. These numbers are not too bad, right? 11 in a million, like 1 out of 10 to the power 5. Now let's see the number of doses the countries have bought of these two vaccines. This is AstraZeneca. This is Himalaya, which is Johnson & Johnson. So this times million. So 1,000 million, 300 million, etc. So these are big numbers actually. So if you take 11 times 1,000 times million, it's a big number. So right now the total number of vaccination as of yesterday is 5 billion. So let's say, of course, a lot of that is mRNA vaccines. But given the number of countries I showed you, a huge number are going to come from AstraZeneca and from J&J. So 11 out of this still leads to thousands of people passing away purely because of vaccine. Something that was supposed to heal you is going to kill you. And so, okay, now let's compare this number to a few other numbers. Just to put things in perspective. Your chances of dying out of COVID is 23 out of million. This is 50% of that. That is alarming, especially in young people. For older people, see the change, 4 out of 800. The chances of getting an injury is 110. The chances of dying in a car accident is 38 and 23. So essentially, your chances of dying in a car accident is only twice, actually 4 times more. still 4 times more than chances of getting, dying out of it. And of course, chances of getting hit by lightning is 1 in. So it's essentially serious. I would say you would consider putting at risk lives of thousands of people and people from the younger age.
So theoretically, everybody should get this vaccine.
Yes, everybody. In principle, if you want to reach herd immunity, 85% of the population needs to vaccinate. And there are, of course, as you saw, countries that are only getting vector-based vaccines. Even India, for example, is getting that. So yeah, it's a serious problem. Okay, $1.2 billion have been invested. Question.
Logically, so what distinguishes, apart from the obvious, what distinguishes a 25-year-old from a 55-year-old?
So,
what makes it more? I mean, is the difference between 4 in a million and 11 in a million hidden in the noise, or is it like there's something really physiological that explains this difference?
No, that's actually a good question. In fact, this is related to the second part here. That what is to do with the physiology of, let's say, aging cells with younger cells. So now the question is, like 11 out of what is physio? There's a lot of things that can be physiologically different. I cannot have one very clear answer for this. But I think one can start thinking about answering this question from an immunogenicity perspective. Like, why is, I have an answer to that, but I'll leave a bit later in the talk to answer this. Let's put it this way. The kind of response, this binding that is happening, this binding can be more feasible in certain kind of cells over another, in people of certain age over another, which would allow this risk to be transcended here. So why is that? Give me a little point. I'll show a hypothetical mechanism where which might answer your question. But the answer is not straightforward. Like I can't give you an answer in the first place because there are so many things. This is of course like this is more like a survey that people have done to find this. Exactly what is the biophysical reason or the real medical reason of why a 25-year-old is more prone than a 55-year-old? That is not very clear. Definitely not in this list. But I'll give you an answer to this. Give me like 15 slides. I'll get back to that. Okay, so now then, the driving research question 1 is how are platelet factors or PF4 functions related to vector-based COVID vaccines? Like, that looks like actually 2 very different things. How can a vaccine have an effect on PF4? PF4 is used when you get cut, and that PF4 comes in to save you. How can a vaccine be there in that? How can it be correlated? So, okay, to do that, the first thing that we did is we determined the structure of this of AstraZeneca's vaccine, we call Oxford vaccine, or chimpanzee adenovirus vector using cryo-EM. So this was a work that we are very, very happy because the experiments are also done in the lab. Not only the computations that we are very, very happy to do. So Ryan, for example, who is a part of our center in Arizona, the Center for Applied Discovery, he worked very hard to kind of come up with particles. If you see, we have close to 8,000 particles. So 8,000 as a number seems very, very small, but this is a P25 capsule. It is a huge amount of symmetry. So you can take this number and multiply this number by close to 4 or 40. And so you actually end up getting effectively at least 32,000 particles, which is a respectable number. So then from there, we constructed the entire capsid at 3.1 angstrom. So this is a minimal subunit of the capsid. So I'll keep using the word capsid and vector interchangeably. They are the same thing, the outer cover, the protein container, so to speak. So of course, once you have the minimal subunit, the rest is using the symmetry and reconstructing the entire object. And so we have been able to find. Let me look into a bit of details of this protein of this system. So now, of course, the cartoon that I showed.
Kind of resolution for this.
The 3.1 angstrom. Yes. And just to put things into perspective, the last structure at 26, which is the Johnson & Johnson vaccine, it came out in 2017. it was 3.7 angstrom. So we will push that. Actually, what happens is beyond a particular point with prior data processing, even squeezing 0.5 angstrom becomes a challenge.
Yeah.
And often you lose some information to get to high resolution. You are actually compromising some, I'll show you. You have to compromise certain parts of the system to improve resolution on another part of the system. The dramatic thing now is an object that I'm showing you as a cartoon right now actually now has a structure, molecular structure that we able to do. So this is of course the outer part of the exterior part of the protein. So these are what you call again the hexon proteins. It has 6 subunits. It's difficult to see here. There are of course something called a thick silicon bundle. This was not completely resolved in the older structure. So for the structural biology community, a lot of new pieces of information are found at 3.1 angstrom, which is not good for. Again, on the inside, there are a lot of small bits which actually can be dissolved in these structures, which are not known before. At 3.1 angstrom, we're trying to, we have gone down to 2.97. We're squeezing very, very hard on a number of parameters. But again, that's possibly at the level of just giving a better number. The biophysical information is more or less very, very similar. Sidechain, and as I showed in the previous slides, we're getting good sidechain information also. Yeah, question.
This was a naturally occurring virus, or was it engineered in a way?
This was it's a great question actually. So this is a naturally occurring virus. So it was engineered, of course, locally at Mayo Clinic. So Mitesh, I forgot to mention, who actually got a DP to an NIH Innovator Award. He has a platform that can actually generate these sort of vectors. So he actually has a vector design / engineering platform, which we actually benefited from. So literally, of course, this is what exactly actually did. Just like absolutely fine.
So the engineer will have it like for the specific purpose they want. I don't know.
They can, but this is the wild type. In principle, they can, and I'll show you that engineering will be done on this.
Okay. And then the proteins which are inside this virus, these are also naturally occurring?
Yes. These are very much naturally occurring. A lot of them are known to interact with the genome, and therefore they are very, very important for the overall assembly. And in fact, these proteins had to be there, otherwise our object is not going to be very stable. And you know our ambition, right? When we see something like this, we think of 250 million atoms in the first place. Right? So we want to simulate this. And if you have an incomplete model, then we will not be able to see. And fortunately, because a computationally interested group is pushing the experiment here, we will do any sort of thing, go to any extent to kind of get the models good enough for computations. And this is all done using MDFF, so the fitting was done using MDFF. A part of that was also using Isoliday, which is like MDFF in OpenML. But because they needed some interactive fitting, but this is all modular dynamics. So, okay, we have gotten a structure. Good. We haven't, so these parts were very, very low resolve. I will not cover it in the talk today. There is separate crystallographic work that was done in Cardiff University in the UK to figure out these parts separately. This entry mechanism of the virus has been studied. We studied that. I'll not report that. It's there in the paper that's gone down. But today let's do something even more fun. Okay, so this is okay. This part is good. So now then you're in a position to compare AstraZeneca's vaccine and Johnson & Johnson's vaccine side by side. This is a structure PDB ID 5PX1. Our PDB ID is to be released. I think we have gotten a number 70 something. In fact, I should have it with my carelessness, we have gotten a number. So what you see is the electrostatic potential of both these side by side. The first thing that you see is like most of the outer part of both these capsids are highly negatively charged. There are of course certain positive pockets. You can see them in both the cases, in both the Ad26 as well as the Charox, but primarily negative. So now of course I showed you a protein PF4, which is highly positive. So the next thing is of course to, this of course experiments were done, but I'll present to you in a way that the computations were done first. So this is of course PF4, and as I showed you, now this is the electrostatic potential on the surface of PF4. It's primarily positive on one side and mostly neutral to marginally negative on the other side. So it's really like meant to bind on dominance. It's absolutely meant to bind on dominance. And therefore that's what, so Chun here did a beautiful Brownian dynamic simulation. When it runs, it should be running anytime soon. Yes. So this is suspect for bit again. So we have run 400 copies of 2 microsecond Brownian dynamic simulations. This is of course the wild type capsid. This is these are the PF4 proteins. These are of course independent simulations. These proteins are not interacting with each other. We're showing them together. Of course, in ARBD now, Alec has code and underlying theory that can actually allow complicated binding. We haven't done that yet. This is all like 400 copies together. After doing that, then we can cluster.
Abhi, I'm sorry. Can you elaborate on how these simulations work on this?
Very good. So, first step is you construct the facet. In principle, when you have a lot of time, computer, not just computer time, because this is also, as you can understand, a piece of work that is going live. Like literally, we have to. This is a real-time response. We chose a facet, which is like 1/3 to 1/4 of the capsid, not the entire capsid. Then we do a little molecular dynamics simulation to make sure that it is minimized. It was already done with MDFF, so that there were no clashes and all that. They were not all there. Once you have the molecular dynamics done, you have to compute 2 things. You have to compute the so Brownian dynamics. It's you need a force field also of Brownian dynamics, right? Because there is a derivative. You see the Brownian dynamics equation. It's a d phi dt equals df plus noise. D phi dt is the rate of change of a collective variable with respect to time. In this particular case, let's say that the collective variable is some orientation or some distance. D is diffusion coefficient, which we measure using hydroprobe, a tool that is available. You give the protein there, and like basically it figures out what is the diffusion coefficient. F needs to be there. For F, which is the force, you need a derivative of a potential. So you create 2 kinds of potentials in Brownian dynamics. As you can see, it's rigid body. So you don't have our regular bonds and those values. So you simply need 2 kinds of potentials: van der Waals and electrostatics. The electrostatic computations are done using APPS. So we have DX files. The van der Waals computations are done using implicit ligand sampling. And so, what's the probe radius tool for ILS? You remember? What's the probe radius?
You need the one solution for the.
Yeah.
For this, I think I use 0.5, but I changed it to other flavor.
So basically, it's a fudge parameter, but depending upon and I'll tell you how to choose the fudge parameter. Sorry to ask you that question. There's a particular width of the implicit ligand sampling to create a van der Waals map, and then you basically take a derivative of that as a function of your order angles, so to speak, to get this. Your noise is actually, I think, fluctuation distribution, and that's what is very well coded inside the and then of course, Chun knows how to use it very well right now. So and so once we have the ARBD running, we find certain hotspots on the surface. And then these hotspots use a particular kind of a model called a Gaussian mixture model to do the clustering. So that what all conditions are finding, that we have to understand. So we actually do that, and we find some very some at least 5 or 6 poses. Most of these poses are on the hexon protein. Remember, I told you there are 2 types of proteins: penton proteins and hexon proteins. Most of the binding is happening on the hexon surface. And we have this, these are some of the regions, more than 50% is red. Like then we have like light red. Somewhere the reds are around 50 to 20, and then 1 to 20% of probability of residence time is in the blue. So primarily on the tips of hexons, 1, 2, 3, 4, 5, 6. You have like somewhat symmetry, not extremely symmetric. I'll break sort of a binding that is happening. And you can see that here. So this is the wild type. So this now tells us that okay, there is some sort of a binding going on. Okay, so now van der Waals interactions. How do we smooth the van der Waals interactions? What parameters should we use? This was actually a work that I did in my PhD, where you should take a protein, just a single protein out of the capsid, and to measure its root mean square fluctuation. This is actually an epitope of a single protein in the virus. If you just take the protein, the RMSFs are very, very high. Now you take the pentamer, not the protein. The RMSF comes down, but it is not an inertial effect. There is actually an velocity inside inside these sort of proteins, which is really, really when this helix moves, these regions on the top are allowed to move. There's an velocity through this. Is an entropic velocity that we have shown in this paper. In a full pentamer or capsid, these parts of the proteins are frozen because they are kind of interlocked with the neighboring proteins, which actually brings down the movements or the RMSFs of the various loops on the surface. And mostly these numbers are sub 1 angstrom. That's what it ends up becoming. The same loops on the surface of a virus, when you take the protein and simulate it separately, its RMSF is a lot higher. But on the surface of the virus, it goes down. And so then this is why you use those kind of numbers that Chun was mentioning, like 0.5 angstrom.
So these are the fluctuations of the same part of the protein,
yes,
but in different contexts.
Exactly. So, and it is one could have said, okay, fine, that a pentamol is heavier than a protein and a capsule is heavier than a protein. It might be some inertial effect, but it's not actually. We found that it's purely an allosteric interaction between the lower part of the protein. When this part moves, because of an allosteric interaction through these beta sheets can actually incorporate entropic allostery. Allostery is mostly enthalpic. You need large scale changes. Entropic allostery happens through fluctuations. You need something very stable, very rigid to do that. Beta sheets have been implicated. So therefore, we find that this smoothing of sub 1 angstrom comes from an analysis like this, which we actually had experienced before. So with that, then, very good. So we took those structures, and if you see now, each one of those clusters, so then Chun got around 22 clusters. From those 22 clusters, we did 30 nanoseconds of NAMD3 per pose. So NAMD3 is very, very effective there. We couldn't have done this with NAMD3. It runs beautifully on GPUs. The system size is small. So thank you for NAMD3. So then we have at least 600 nanosecond. So yeah, we have at least 600 nanosecond of sampling per pose, so to speak. Then of course now we have to save time. I cannot do the FEP calculations that we mostly do. So we resort to scoring functions. This is available on a tool called Prodigy. It's actually very, very useful. Like you actually have a normal installable interface. You can do it on your computer. You don't have to like put everything on the web server. So you just upload your poses, the local poses. Here is a negatively charged virus surface with a positively charged protein on it and build on it. And then you find this KD values, which are of the range -6 to -15. Let's take a mean of this. Medium, somewhere in the middle, like -10 kilocals per mole. This is of course Chadox. This is AstraZeneca vaccine, and these numbers extend the same range for the Johnson & Johnson vaccine. But the Johnson & Johnson is a tad bit lower. It's not very low, but it's in the ballpark. So we have been able to then get some sort of KD values. Oh, between this, of course, we did M. So then we did BD. From there we did MD. And then we used this KD values using the Prodigy server. So now we actually have a knowledge of how strongly does PF4 bind to the capsule. Good, but these are computational values. Shouldn't trust that. So then we did experimental validation. Number one is surface plasmon resonance. So in surface plasmon resonance, what you do is, as a function of time, you basically, these are the PF4s. You actually flow the PF4 on the top of an immobilized capsule. And then you measure what is the response of the surface plasmons. Basically, when the binding happens, then your this response increases. And after a certain point, when the surface is completely full, then unbinding starts. Unbinding of the PF4, and this goes down. So basically, you see at different salt conditions, 150 millimolar, which is our normal salt condition, this is a much stronger binding. Now, if you keep increasing the salt concentration, its interaction is coming down. So it's basically an electrostatic interaction. You're increasing the salt concentration, you see it's treating it. You can actually, so this, because it's time dependent, you can get K on and K off values out. Definitely you can get KD values, the dissociation constants. And we found like these are of the order of 600 nanomolars for Chadox 1, 300, a bit less for Ad26, Johnson & Johnson. Very similar in terms, when you convert that to kilocals per mole, you get very similar. So we're very, very happy to see that. Let's start with Brownian with cryo model, followed with molecular dynamics, Brownian dynamics, molecular dynamics. And then doing a KD calculation, we found certain numbers that were very, very close. And these experiments are not even done by us. This was done in AstraZeneca. This was done in the company. So when they found these numbers, that's when they kind of started believing us. Okay. So this is the first time we're getting somewhat towards the mutants. Okay, good. So next thing is okay, fine. So now we have this is the PF4. So PF4 is often found in isolation or with a heparin, which is an anionic polysaccharide.
Sorry, Abhi, yeah. Your conclusion from the first delta G and KD plus SPR measurements is that AstraZeneca's capsid binding more to PF4?
Marginal.
Margin. Okay.
Yeah. As opposed to the fact that this doesn't bind at all.
Yeah. I see.
Because originally the thought was that it won't happen.
Yeah.
Because in their normal trials there was no blood clotting event at all. Okay. So this still I haven't given the why is bit happening, why is the blood clotting happening. I haven't told. I'm simply telling that there is evidence now that the vaccine is interacting with this PF4 patch. Next is, of course, the next test is okay. So there is, like as I said, an anionic polysaccharide called heparin, if you bind, which is a natural binder of PF4. So when this joins, the electrostatic surface looks a bit quadrupolar, like basically you have like two dipoles, positive and negative charge. When with this you do Brownian dynamics, you get a surface coverage like this. So from 2,600 hits of those, if you remember, 800 microsecond Brownian dynamics simulations, they've got down to 210. And when people does experiments of this, they hardly see it. So this we can consider as our baseline. If in the future we can design a capsid where the hit is 210 or lower, then we would have gotten a redesigned AstraZeneca vaccine where this problem is over. So we have been able to set our benchmark. Okay, very good. Again, we did the surface plasma resonance. This is a much high binding in the presence of the PF4, and it comes down when heparin is added. So again, these experiments were done in AstraZeneca. So they were, in fact, they are a part of the paper now. We have to go through a little bit of IP and all that, but it's a part of it. Finally, a separate paper came out showing super resolution imaging, where they claim that these green dots are PF4 that are sticking on the surface of the Chadox capsid. But one thing they have shown in plots, they have computed the hydrodynamic diameter. For control experiment, this is the wild type of the Chadox vector of the adenovirus. Then they have added PF4 and its size increased. So as if there's something accumulating on top of it, the hydrodynamic parameter, diameter. This is like essentially what they're claiming is the diameter of the PF4 bound Chadox complex. Then they added a bit of DNA. What they feel is DNA is acting like essentially it attracts more positive charge towards that, and that goes up. And then finally, when they included heparin, the diameter came down. So then this is also like another evidence showing that there is electrostatic association between these complexes. And this is two of them were of course by us. The third one is by separate people. So now then, what is the working hypothesis? So on the day of vaccination, you get vaccinated. This vector enters your bloodstream. Now, inside the bloodstream, this vector is binding to the PF4. Now, by the nature of the vaccine will have to get into the lymph node, where the all the immune response subsequently happens. Not the immune response, actually, the anti-immune response subsequently happens. In principle, a PF4 cannot cross a membrane. This is a very, very important property. Any kind of cytokine protein, by definition, cannot cross a membrane. What we are hypothesizing is this PF4 bound vaccine. The vaccine is not allowing, it's a secondary vehicle. Inside it is carrying the genome. Outside it is carrying these PF4. Now, when the vaccine goes in the lymph node, this PF4 gets in. Once this PF4 gets in, then downstream the body sees, okay, PF4 is in the wrong place. Let's now start generating antibodies, auto antibodies, its own antibodies. These antibodies come into the picture. and then this complex kind of gets made, and subsequently blood clot. So this is a hypothesis that we are following. Now then the big question is, why is this not happening in everyone's body? I'm trying to answer Chris's question. Everyone who is injecting, physics is the same for everyone. Right? So physics should actually allow everyone to reach up to this stage where the vaccine is binding to the PF4. Now, and then also, if the vaccine is bound to the PF4, this will get in. There is no problem with that. So it turns out, that's why I couldn't answer Chris with a straight face. These antibodies that have been generated, these are a very, very rare thing. Not everyone can generate these antibodies. So for those people who have gotten this blood clot, barring those people have done antibody testing. This antibody is not found. So for some reason, those people who are younger are actually generating more of these antibodies. Might be because they're young. So, but I don't know that. I can't say that for certain. One thing I've learned after this project is biology is about diversity. Physics is about unity. And therefore, we always like to kind of simplify things. But life is, of course, nowhere near that, especially when you're taking a responsibility of something like this. You cannot be careless. So this antibody that is being bound, it might be that a younger person is more active, the cells are more active, it's generating more antibody, and therefore getting into more problems. But this is our initial hypothesis. This is, of course, so the paper got accepted in Science Advances. We wanted the advances removed, but of course, the thing is, they didn't allow us a quick turnaround. And we really wanted to have a quick turnaround of this, so we just went with it. Yes, Chan.
So if you have a less negative capsid, then this problem will go away, right?
Okay, wait. So how can we de-risk this vector delivery? I cannot show you this. Now I'm getting into NDA territory. There is a pipeline that we have generated with colleagues at AstraZeneca. Part of it is Brownian dynamics, part of it is molecular dynamics. These parts are magic. So now with that, then the idea is to redesign certain parts, the capsid, so we can actually reduce this. One thing is, of course, in the meantime, a lot of work we're trying to do, actually the Oxford vaccine team is trying to do in Oxford, to figure out the cryo-EM model of a PF4 bound to the capsid. But turns out that the capsid is symmetric, but the PF4 binding is not symmetric. So doing cryo-EM is extremely difficult. And essentially the cryo-EM density is spread all around. You're getting fuzz there. So that's something that.
But didn't you find that the video Brownian dynamic simulations that hexamers on the sites that usually PF4 bind?
Correct, but now when you're doing a cryo-EM experiment, you have the entire capsid plus you basically have highly heterogeneous, conformationally diverse capsid PF4 models. Because the thing is, it's not like all the capsids will bind the PF4 in the same space.
Sure. Right.
Had that happened, things would have averaged out, it would have gotten a very good structure. But that's not happened. So therefore, that's a problem. But despite that, we moved on. And yes, you are right. I cannot tell you what these mutants are. They cost actually. So they're actually very so Chun, of course, has now weekly assignments. So Chun basically works with me, Chun, one of my students, Eric. So every Friday we meet with AstraZeneca and give us literally on a weekly timeline on how can we de-risk this object. And by certain magic mutations, we have now able to get down from 2,638 wild type to 26, which is much lesser than 210, which is remember the baseline bound. So therefore, there are mutants that we have been able to Chun is under NDA, he will not say that has been designed. So that this situation can be done. And we actually now have, this mutant is, people are doing things with this mutant. Okay. So how much time do I have?
So 10 minutes officially.
10 minutes, that's fine, 10 minutes good. Okay, so now then, and this will be, of course, the next paper of Chun and Eric on how did we rescue the vaccine message. Now that we know what the mutants are. Hopefully, and so my contract with AstraZeneca is, I will be giving them this mutant list. I will not ask them which one is findable. I I will not know that. I simply, the model that we are built using NAMD and DMD is our. Yeah.
Do you also look at what these mutants will cause to the stability of the capsid?
Yeah.
How do you do it?
So that's where we like NAMD image. We can't do too much more than that. We're introducing the mutations, doing some sort of local sampling, and seeing if the interface is changing. But the people say that, give us something, we'll do it in the experiment. It's so much easier to find out the stability in the experiment. And like, if one candidate fails, so I'm showing mutant 1, mutant 2. June actually has how many? 12, a list of 12 mutants that he's testing. And so using Brownian dynamics, we're also doing it parallelly on our DGX2 and NS2 and on our other nodes. So everything, like we have put all the project people who are not working on this project complains a lot in the group right now because all the computers are taking on this. So, but we're pushing this very hard. So yeah, even within the molecular dynamics, we find that it hasn't compromised as much. And of course, that's also a part of when I was showing you this hidden protocol. Some of that is there also, which I cannot say too much about the things under the blue box. So okay, good. So then now June has rescued us. Very good. So if I have a de-immunized, let's say a de-risk AstraZeneca vaccine, is that the end of the story? Is what else can cause trouble? So let's take you back to another company's work. Merck had also started pretty early in this area, and they gave up. I think this was around like early this year. They came up with this small press release. They say that after their finding, this was generally not too bad, but the immune responses were inferior. So you have designed a vaccine, great, you have designed a vector. The vector is not causing any side effect. Of course, it was not supposed to in the first place. It was causing possibly risk. But will that still be a good vector? Let's think on it. Let's say I'm using a vector of a particular virus. Okay? That virus has infected me before. I already have, my immune system is already trained to not recognize that virus. So if I was introduced to the ChAdOx vector, somehow, my immune system knows that vector. When I'm using the same thing as a booster dose, for example, I didn't tell booster dose, but let's say as a third dose, and then I introduce this, my body is already immune to this vector. It would not even let this thing get in. That's a big problem. So now it's a problem of pre-existing immunity. If I am immune to a particular vector, you do whatever with that. My immune system is not going to let that thing. So now even if I have a de-risk vector, do I have a de-immunized vector? That's the next question. And that's what we are trying to address in the last 6 minutes. Okay, to do that, we developed another tool. It's called Ensemble MHC. So, so basically, as I was telling someone, like this VITT, pardon my French, but I didn't know SHIT about this 1 year back. So so again, this is another protein. It's called the MHC1 complex, major histocompatibility complex. So Eric, again, he figured out a relationship that these complexes are present in people from different backgrounds, from different countries, in different, so there are different variants of these complexes that are popular, let's say, in the Caucasian population versus in the Asian population. And based on what this complex is, you can actually basically rank the country according to their risk of death. Lower the number, the more risky are they in terms of. So he came up with a score called the EMP score, which in fact we have factored in the Delta variant also. And then there are countries where the death will never be as high. It will, even though there can be high infection, the death will not be as high. This was, okay, it's a prediction, of course. Right now, we are actually overestimating the death by 10-fold more. The sheer number of deaths, because we have not included vaccination in this model. We included a lot of other things like age, gender, but not vaccination. But still, the trend is very similar. Like countries that are on the lower part are actually more prone to death. and that on the upper part are less prone to it, because people from these countries have different kinds of MHC1 complexes in their remote in their genotypes.
What is changing when this thing changes?
When this thing changes, it's basically here.
Oh, the day. Yeah, I see.
So now then, this object that you see here is actually an epitope. So it turns out that this huge, beautiful object that we use to determine the structure using cryo, there are parts of the cell that doesn't care about that structure at all. So what happens is, I should have introduced the problem better, but let me just do a hand-waving introduction. Once this capsid enters the cell, it does whatever it does, then it goes through this thing called the proteasome. The proteasome is like a wood chipper inside the cell. It takes an entire capsid and pieces this into multiple 8 to 14 peptide chains. So it turns out a SARS-CoV virion with all the proteins in it, it will be able to construct 14,000 to 16,000 peptides. So these peptides go on and bind to the MHC complexes, which are presented on the cell, which is now, like this presentation actually triggers downstream long-term T cell immunity. So it's not the B cell, which is the short-term immunity that we're talking about. T cell immunity is immune memory. So it turns out mRNA vaccines have a very good long-time immunity. So basically our long-term T cell responses are intact for a very long time. So okay. So now then, what we came up with this tool does is, okay. What this tool does is it's a bioinformatic tool. So you take this object, split it into 14,000 to 16,000 peptides. But turns out for each one of these peptides, there is mass spectrometry data that is available on whether given an MHC, this peptide can be presented or not. That is known. So we actually score these peptides by a product of how strong does it bind to the MHC complexes times is it presented to the TCR receptor or not. And once we have that, from this 14,000 to 16,000, we come down to 658 peptides, which can actually be presented by the SARS-CoV-2 virus. Now, let's say this is one of the MHC protein. This protein is primarily found in the hemoglobins. This is another type of protein. This is found primarily in Asians. The reason I have chosen 52 is because I have 52 crystal structures that is available. In total, there are 400 such proteins that is available across the world. So based on what protein you have, this software allows to tell you, with which epitope with someone from a European genotype bind versus with someone from an Asian genotype. And based on that binding, we can predict whether that person is going to, we can correlate first. Whether that person is going to be, so okay, two things. First of all, we find the binding. Then we go to the Johns Hopkins database and figure out, so it turns out that every country has a very strong MHC database. If you go to any country, like in India, for example, people have given blood. And there's a database that tells that, how many people have this kind of a MHC protein versus this kind of MHC protein. Then you go to the database in Johns Hopkins. And then you ask, in this country, how many people have that? Then you have 1-2 data points. This country has this MHC. This country has a certain number of deaths. Then you can actually correlate them, and we found a linear correlation. This was not found before us. So this was actually something that we found very, very interesting. So then, of course, we can now map, because you know the structure, we can map out where exactly this epitope lies, the red regions. Okay, so this was a tool, this ensemble MHC of determining where the T cell epitopes are. Now let's use this with Javascript. So we take the same T cell epitope determination tool and figure out what are the where are the epitopes that can bind very strongly to MHC complexes. Turns out, again, like there are I think 69, like a total of let's say 70,000. It's Shadox. It's much bigger than the spike essentially. More number of peptides, not 14,000 to 16,000, but like 70,000. most of them are very, very weak binders to immunity. Forget bonding. We see that there are only 46, 1% of the total population that needs to be, those are the epitopes, which is what is going to cause our body to recognize that this is a Chadox. If we somehow cover these epitopes, cloak these epitopes, then we can use the same vector. So the vector, first of all, we de-risk the vector. We remove regions that would have caused PF4 binding. If in doing so, some of these epitopes get covered, well enough. If not, we are down to 46, around like this region, which still needs mutation, more mutation. So then we started doing computations, and then we made a big number of mutations. Fine, whatever the good things are, let me show the bad thing, and then I think I'll stop. Turns out some of those epitopes, which are cross, which are basically which needs to be cloaked or covered, are actually at the interface. Mutating them might affect the stability. That is where we're starting. So despite the fact that we have potentially de-risked the vector, which is experimentally tested, we haven't yet de-immunized that completely. Because some of the epitopes that can be re-engineered, most of them are on the surface, it's good, but some are at the interfaces of the proteins. And here mutations needs to be done very, very good. So yeah, that's why we are stuck right now. That's our impending challenge. So with that, yeah, let me just tell you what I showed you today. That the first oracle model of Chadox has been shown. PF4 binding to Chadox1 can induce this side effect. We have been able to show that. We were at least able to hypothesize that. We also did a lot of work on PF4 entry, which I didn't show. Let me show you another trial map where you see this thing sticking out. These are what you call the fiber molecules, which I didn't have time to show you, but they are resolved separately. Can we de-risk the vector delivery? Possible. We have designs, they are tested. If that comes out, then possibly AstraZeneca moves in a particular direction. But can we exist, work in progress, pre-existing, can we do anything? So I don't know. We are working on it. So yeah, that was the work for the last, I would say 6 months. So yeah, with that, I'd like to thank you and thanks for that.
Very, very exciting episode. I can't tell you how happy I am that we are here and you are here. This was really, really nice inaugural for our TCP seminar again after the pandemic. For the first time, class actually at the end of the seminar.
Thank you.
Well, I mean, the forum is open for the questions, also the people who are online, so you can just turn on your microphone and ask. So you don't have too many people actually to worry about. But let me actually start with a comment and a question myself. So this negative positive protein, heparin, and this business seems to be also very important in blood clotting, pointed out. So some of us are working on the clotting factor.
Right.
And we have really positive and negative centers in these proteins. And that's why heparin works against those proteins.
Yeah.
They need to find negative lipid and get activated. So that's actually a very interesting area. But my question was the following. So you seem to have studied the vaccines very much. So my understanding is that the mRNA-based vaccines are the most effective one, more expensive, of course, and it's difficult to produce them. But why are we still sort of investing so much in the other types of vaccines? Isn't it the case that maybe, I don't know, 3 years down the road, everything is mRNA-based and we are done, we don't have to look at the old models anymore? What is your...
I think the verdict is not out yet, but it is true that even companies that have invested in vector-based are thinking of technologies that are mRNA-based. It's an active area, but there are expenses. One thing, for example, as I was discussing with you yesterday, some of us might have taken, I think, Pfizer or Moderna. You need cold chambers, fridges, for example, that needs to be stored. Go to Africa, and you need those kind of cold lines. So therefore stability, storage is distribution, ways of mechanisms of distribution is one thing. So, but I think the reason people like RNA is because a lot of medicine now is working towards personalization, and control on the genome gives you that handle. Right? Like for example, I was showing you countries, how different people from different countries on a broad level have can be classified using a certain kind of protein. Now, if you want to make a very targeted vaccine for people of one kind, which possibly will help them more than others, the only way of getting that kind of a control is by getting a handle of this. So I think that's the reason it's very, very exciting to do RNA.
So any other questions? Oh, yes, please.
Yeah, so in the first part of the talk, we talked about PS4 binding. So what I got was electrostatically driven, but there's a lot of other electrostatic particles in our body. So what gives it the specificity, or have you looked into that?
I think no, that's a good question. So first of all, it's correct that positively charged objects remove. How many of them are in the bloodstream in the first place is the first question. I cannot give you an answer because I know that there are other factors. There are other proteins that are now looked into. But I think the bigger question, the reason PF4 was, if I go back, the reason PF4 was thought of was not because of this slide. It was primarily because of this slide, where people actually found PF4 antibodies in the patients where blood clotting has happened. Had that not been there, so the causality of the thing is when blood clotting happened, people looked into what is the reason, and they found PF4 antibodies. And then they went back and started looking into what can happen. This as I said, diversity, right? A lot of things can happen. So you cannot, even an experimentalist or any big biopharma, anyone is following a model. So the model comes out of data. When blood clotting happened, they found that PF4 antibodies were present, anti-PF4 antibodies were present. And that was the reason they backtracked and say, how can that happen? And then they found that, okay, if this route is activated, then anti-PF4 antibody can happen. And therefore the PF4 was activated. Are there other things that can be found? Are there other complexes with which this thing can bind? The answer is yes. But despite that binding, will that lead to this sort of downstream events? Barring one such protein, which I can't name, I don't think in the conversations that people have been having, and that other protein looks exactly similar as PF4. Like if you're interested, just do a blast search with them. Or do AlphaFold. People have started using AlphaFold, right? Just do a little bit of AlphaFold. You find like a couple of proteins that look very similar to this protein. One of them has actually been implicated in some of the recent meetings that we are getting into. But yeah, so that the answer to your question is, it didn't start from the fact that which protein should be there. There was a measurement of the antibodies. They found that PF4 antibodies are present and they are polluted.
Yes, sir.
You mentioned that the PF4s primarily bind to the hexamers. Why was that the case?
Well, it was according to that picture, which was there in the paper because we were doing a smaller facing. It actually also belongs to the pentamers, at the interface of the hexons and pentam. Well, basically, it's like, answer is simple. Whenever there's very strong local negative binding pocket, you'll be able to get that. And because it's not symmetric at all, statistically speaking, if you take one vector, the binding pattern on that might be very different from another. So when you put them all together, you'll see that the entire surface is covered, which is what was shown actually in the super resolution image, which is somewhere here. So essentially, this kind of a coverage. It's as you see, it's very asymmetric. So it will definitely also bind to the. It's easier to make a model to start with exon and then you graduate.
I see.
As they say, more the comment than the question. It seems that now your workflow that the team developed has to be included in the development of vaccine.
Yeah. Yes, that is correct.
Okay, Ali?
Not a science question, but an outreach question, actually. So the words vaccine and risk are misassociated all the time among the public, so much so that misinformation has itself become a disease vector and is doing almost more harm than biomedical conditions in some communities. So do you see a strategy based on either the mechanistic explanations or the statistical studies that you have access to that we can use to reach those that show vaccine hesitancy to great detriment?
Yeah, like this is
basically, is there a narrative we can take perhaps from your advice?
So I can tell you a project that we are doing, right? So you know, like we have also education initiatives as part of the NSF career. We have to do some of this. So what we are doing is we're teaching high school students how to re-engineer this chemistry on VME. So basically, you have the mutator tool on VME. You can take and like as Jalu suggested, just build like you take the mutator, change the surface, and make electrostatic potentials. Very simple thing to do. Essentially, might be when the entire class does that, this magic recipe that I was putting under the blue box will be gone complete. So because some answer will be right, and like not. So now the question, yeah, something like that, where you engage people on a virus in a computer. So in fact, when we talk about viruses on a computer, the first thing people used to think is like, do you not have Norton or something like that? So I think, yeah, something to that extent, which we do, but I think beyond that, it's a question that is not, possibly addressed.
Check one more time. Any question from online people? You can just raise your hand or type your question or just speak. If not, okay. Okay, well, I mean, thank you very much again. I mean, this was really powerful and insightful. I think it's a good lesson for your students. So how much more data is out there that we are not tapping into? Besides how I've connected the molecular simulation results to how it's bioinformatic information about 16,000 peptides coming presented by the cell to the surface and interacting with MHC and so on and so forth. So if you can combine actually your molecular simulation to any outside database, then the study becomes an order of magnitude more relevant to real biology. As I said the same thing during my investor chat. Biology is complex. I didn't use diverse, but as Abhi said, biology is complex and we can only capture a very, very small part with our simulation. It's always a good idea to go out and connect to more real kind of presentation. Thank you very much.
Thank you.
Okay.
Yeah.
Thanks.
Just to coordinate. So I'm going to pick up Teja. I don't know what your plan is. I want to make sure you're not stranded. Okay, that's what I was thinking. So I assume that Ahmad is going to give you a line.
Yes, so in the first one, worst comes to worst.
You can always call me.
No, I'm happy to.
I know you can, but I don't want to leave you in the.
No, it's very nice of you, but again, if I'm stranded.
Then I'll just see you tomorrow.
Thanks for checking. Thanks for checking.
This is perfect. We should do more of this. Of course. Thank you.